All right. Well, I want to welcome everyone again. It's Mitchell Warren from AVAC, and delighted to welcome you to now the fourth in a 5-part series in advance of HIV Vaccine Awareness Day, which, as I hope everybody on the line now knows, after at least after these calls and updates, is next Thursday, May 18th, a week from today. And we are delighted to have today's call, which, frankly, of all the calls, is one that we might not even have imagined 28 years ago. And I'm going to explain that as I introduce our guest, Lynn Morris, in just a minute. But first, just a couple of logistics issues. We are keeping the line muted so we can record this. It'll be posted along with all of the other calls on the website. But there is going to be a chance to engage and ask Lynn questions directly. And we'll be able to, I'll give you instructions on how to unmute your line. But throughout the call, and as you hear Lynn present, you're welcome to add your questions to email us at avac@avac.org, or you can use the ReadyTalk chat feature if you're following along online. And that is kind of the run of the show for today. I do want to just remind people that if you do go to avac.org/HVAD, you will get the recordings of the previous 3 calls and soon the recording and slides from this call. You can also register for next week's final call in the series with Nelson Michael of the Military HIV Research Program, as well as a range of other materials and resources, some of which are already updated in there, including one about broadly neutralizing antibodies, which is an advocate's guide and a very, hopefully, user-friendly way to navigate through and understand some of the concepts that Lynn will describe. And there'll be other materials that will be continually loaded there over the coming days in advance of next week's HIV Vaccine Awareness Day. For those of you who were on last week, I guess almost exactly a week ago, with Barney Graham from the Vaccine Research Center, he described some of the history in terms of the speech on May 18, 1997 from Bill Clinton that really launched the idea of HIV Vaccine Awareness Day, and then provided an update on the Vaccine Research Center, one of the outgrowths of that speech, and a really important center of excellence. And Lynn, I think, will even talk a bit about that in her talk today. But as I say, Lynn's talk is of the 5 that we have, probably the one that would be the most unknown in May 1997. You've seen 2 calls now. The very first one with Maria Pau from Janssen talking about a new vaccine program, the AD26 program that's going in possibly to a large-scale efficacy trial later this year or early next. Just earlier this week, you heard from another South African, Fatima Lahare, about the efficacy trial that's already going on in South Africa, the HVTN 702, classic vaccines that have been developed over many years that are in trials. And those were the things that we all thought about and longed for, not only in 1997, but for 35 years, and as long as we've had the epidemic. But as Barney described last week, in addition to the Vaccine Research Center leading vaccine development, he described the beginnings of the work related to broadly neutralizing antibodies. And we're delighted that Lynn Morris is here today to do an overview specifically on that. And I think if many of you on the line will know that if one looks back over the decade or so, or even 2 decades, the enthusiasm and excitement around broadly neutralizing antibodies, the discoveries that have been made just in the last few years, have really provided amazing opportunities for the field, but always, as we know with HIV, challenges as well. And Lynn is going to unpack all of that in her talk today. We have the fabulously provocative title plan bNAb and an update for broadly neutralizing antibodies for prevention. And I'm delighted to invite Lynn, who has been a pioneering researcher in South Africa. As many of you might know, also she was one of the co-chairs of the most recent HIV Research for Prevention conference in Chicago last October, and provided leadership not only around all things antibody and vaccine, but pulling together a broad conference related to HIV prevention writ large and where this all fits in. She actually spends her time mostly, though, in South Africa, where she is at the National Institute for Communicable Diseases, and also works very closely, as you will hear, as part of the CAPRISA team, the Center for AIDS, for the AIDS Program of Research in South Africa, which has done pioneering work across a number of prevention and therapeutic areas. So, Lynn, I am delighted to welcome you, and we'll turn it over to you. And then, again, please, people, send questions in the chat feature or on email, and we'll open the lines when Lynn is done presenting.
Great. Thank you very much, Mitchell, and welcome to everybody on the line. And thanks, Mitchell, for this provocative title, actually. I think you gave me the title, which I think is great.
Well, that would be,
and of course,
I just want to ask you credit to
of course, Plan A. is to get a vaccine.
I get that.
But now that we have all these broadly neutralizing antibodies on hand, we are testing them for prevention, and that's what I'm going to describe today. So if you could move on to the next slide. So I think, as you all know and you heard earlier in the series from both Fatima and from the Janssen talk, that we really are at a pivotal point in the HIV epidemic. We've got 2 efficacy trials in the field, and a third one, due to start in the coming months. And just to say that both of these, the current 2 trials that are going on right now, both rely on HIV-specific antibodies, but in very different formats. So on the next slide, you will see that the more traditional vaccine approach is what we call active immunization, and that's what the HVTN 702 trial is designed to test. So this is a classic vaccine which people are immunized with, and the idea is that it stimulates the host immune response. And in the case of HVTN 702, the types of antibodies that we're looking to elicit are binding antibodies, particularly to a part of the virus known as the V2 region of the virus. Those are what are called the correlates of protection. That's what we're looking for in that vaccine. But what I'm going to talk about today is passive immunization. So this is where we infuse antibodies that are made in a laboratory, and these are infused, and I'm going to talk specifically about the AMP trial, an antibody called VRC01. And the idea here is that this provides, that these kinds of antibodies will be able to protect somebody from HIV infection, and that's really what AMP is aiming to test. So if we move on to the next slide. So as you know, we have vaccines against many viral infections, and most licensed vaccines, we know they work by inducing neutralizing antibodies. So these are the kinds of antibodies that basically bind to the virus, and they prevent virus infection. So they directly inhibit or neutralize the virus. And as you all know, vaccines are given in early childhood generally and provide lifelong protection. If you move on to the next slide. So through many years of study, we know that some people who are infected with HIV naturally make these kinds of antibodies called broadly neutralizing antibodies. And there's been a lot of different groups, spending a lot of time identifying people who make these kinds of antibodies and, of course, isolating them and studying them. And they've been really crucial for both for vaccine design, but also, as I'm going to tell you, for the passive immunization agenda. So these so-called co-evolution studies, where you look to see how the virus and the antibody basically chase after each other. There have been a number of these co-evolution studies that are really telling us the kinds of stimuli that the immune system needs in order to make these kinds of antibodies. And of course, that's what we want to do by vaccination, is get the body to make these kinds of antibodies in a regular way by vaccination. But as I said, they also are a source of what we call biological drugs or biologics. These antibodies can really be used as PrEP, essentially, for passive immunization studies. So on the next slide. So these over hundreds of these antibodies have now been isolated. And what they've done is that have allowed us to define the sites of vulnerability, so the soft spots that we want to target on the virus. So what this is here is what we call the trimer. It's the protein that surrounds the virus, and that's what neutralizing antibodies bind to. And the colored sites are the so-called broadly neutralizing antibody epitopes, as I said, the vulnerable sites. And you can see that a lot of this trimer is really covered with color. So a lot of the trimer is actually a target for broadly neutralizing antibodies. And in fact, we have good antibodies to all of these sites. And the one that the two that I'm really going to focus on today is the one in green, which is the CD4 binding site, and that's what VRC01 recognizes. So this site on the virus, this is how the virus interacts with CD4 on the cells. And so obviously if you block that site, you're going to block virus entry. The other site that I'm going to focus a little bit on is the V1, V2 apex site, which is colored in red, which is right on top of the trimer. But I should say that, you know, there are very many good targets on this trimer. And if you move to the next slide, we'll see that antibodies that target these different sites really show differences in what we call breadth and potency. So, as you can see on the bottom, the breadth of coverage, as you can see, it goes all the way up to 100. So ideally we want to neutralize 100% of viruses. And then on the left-hand side is what we call potency. And that just tells you how much antibody you need to neutralize. And so as you can see, those numbers get smaller and smaller, meaning you need less and less antibody in order to neutralize viruses. Now, ideally, we would like to be in the sweet spot, which is you have an antibody that neutralizes lots of viruses at low potency, at high potency, so at low concentrations. Indeed, there is a very new antibody actually that's getting pretty close to that sweet spot. It's not on the slide because it's a very newly isolated antibody called N6 that was isolated by the Vaccine Research Center. So I guess that just also makes the point that people are isolating better and better antibodies all the time. But the ones that are on this slide, many of these are also going forward for clinical testing. So you can see, and I've also color-coded them the same way that they were color-coded on the previous slide, with green being antibodies that target the CD4 binding site. And you can see VRC01 is hitting around 80% of the viruses. But it's low on the potency scale. And you can see that there are other CD4 binding site antibodies that get better in breadth and potency, and certainly the VRC07 is also going forward for clinical testing. Then if you look at, there's a whole, all the blue and the red viruses tend to kind of all neutralize the same, have the same sort of coverage. And I should say that this is very focused on Clade C isolates, and that's because Clade C dominates in Southern Africa, and that's really the focus of the work that we're involved in. But I just want to highlight as well the CAP256 antibody, which, as you can see, is a very potent antibody, and that's one of the reasons why we are interested in pursuing that antibody. So if you move to the next slide. So what are the desirable properties of an antibody in order to take it forward for HIV prevention? Obviously, safety is the primary concern. And I should say that antibodies are being used for many other conditions, not so much for infectious diseases, but for inflammatory bowel diseases, cancer, a number of other diseases, and certainly there have never been any safety issues, major safety issues. So obviously, but safety nevertheless is a very important criteria, and obviously to be effective at low doses. So that speaks to the potency issue. You want a potent antibody. Obviously, it must be broad. You want it to hit as many viruses as possible. But in order to achieve this, we may need to use more than one antibody, and certainly people have done a lot of studies looking at combinations of antibodies. You know, ideally one would want to have an antibody with a long half-life, up to 4 to 6 months, and so that one doesn't have to give injections, well, you only have to give injections every 4 to 6 months. Ideally, for practical purposes, that it's injected subcutaneously rather than intravenously, which is obviously what the AMP trial is currently doing. But the idea is to move towards a subcutaneous injection, which people would be able to administer themselves, and then that the costs are comparable to PrEP, because obviously that's what we're competing with. Now, you know, the cost of goods is always, it's a moving target, and certainly people are doing these kinds of calculations, and that you know, if the demand were there, certainly the costs would be comparable. We move on to the next slide. So you heard last week from Barney Graham about the AMP trial. So AMP stands for Antibody Mediated Protection, and it's a trial that's being done by the HVTN and the HPTN, and it's an efficacy trial, so a phase 2b study, to look to see whether VRC01 broadly neutralizing antibody can reduce HIV infection in high-risk individuals. And It's a study that's been done in the Americas amongst high-risk gay men, and in Southern Africa amongst high-risk women, heterosexual women. And I think when this trial was first proposed, a lot of people were quite skeptical about the schedule, because it's 10 infusions, intravenous infusions over a 2-year period. And I'm very pleased and delighted to say that the trial is proceeding extremely well. In fact, the enrollment is they're ahead of the target for the enrollment. So the trial is going extremely well. So if you move on to the next slide, obviously there was a lot of preparatory work, to do the AMP study. But what the aim of the AMP study is to show that the VRC01 can reduce infection rates in high-risk groups. And one of the really important things that this trial will tell us is how much antibody you need in the blood in order to prevent HIV infection. And that obviously feeds into the vaccine agenda, because that will tell us what sort of levels of antibody we will need to elicit by a vaccine. The other thing is that studies on what we call breakthrough viruses, and I'm going to talk a little bit more about this. So those people who are on the active arm of the trial who become HIV infected, we expect that they will show greater resistance they basically will be resistant to VRC01. And we can actually measure this in the lab, and we can also study the virus and find signatures of neutralization resistance. So the next slide just talks about VRC01, which has now obviously been through a number of phase 1 safety studies, and this is the one from Julie Ledgerwood from the Vaccine Research Center, showing that it was very safe. There were 2 infusions. But the important thing about this study is it gave us the potentially protective levels, which lasted for up to 8 weeks, and that was really what informed the design of the AMP study. And then on the next slide, I've just shown you what this really looks like, because one can measure, after somebody's been infused with VRC01, one can measure the levels of VRC01 in the blood of the participants. So this is data from the HVTN 104 trial, as I said, another phase 1 trial. So on the left-hand side is people who were infused with 10 milligrams per kilogram, and on the right, 30 milligrams. And as you can see, the levels on the right are higher, as you would expect. And you can also see that shortly after infusion, the levels of VRC01 are immediately measurable. And then over the next 56 days or 8 weeks, those levels decay, as you would anticipate. And then the next infusion, you can see that the levels of VRC01 immediately shoot up again and then decay again. You can also see that there are, people do obviously metabolize or the levels of VRC01 in individuals. I mean, they are pretty tight, but they're not identical. So we move on to the next slide. This just shows you a possible scenario. So this is now how we're going to measure efficacy in the AMP trial. And this was put together by David Montefiore and Peter Gilbert, based on a protective efficacy of 60%. So if you look in the treatment group, and what they've done is they've modeled the 10 infusions. So those are the 10 peaks that you can see of VRC01 that are going to be measured in people who receive VRC01. Obviously, there's no VRC01 in the placebo group. And then the little red dots are the infections in the treatment group and the placebo group. And we are anticipating that there are going to be far fewer infections in the treatment group. If you move on to the next slide, what we will be doing in the laboratory is isolating the viruses from both groups, and obviously this will all be done blinded, and measuring the sensitivity of these viruses to VRC01. And what we are anticipating is that the viruses in the treatment group will be largely resistant to VRC01, and that will explain the infections. Whereas in the placebo group, we expect to see a large number of sensitive viruses. We also anticipate that some people will be infected with partially resistant viruses when there are detectable levels of VRC01. So those are the 3 that are still in red. They're not completely resistant, they're only partially resistant. And then that will also help us to figure out, again, the protective titer of the antibody that one needs to prevent HIV infection. If you move on to the next slide. So hopefully the AMP trial is going to provide this important proof of concept. The CD4 binding sites broadly neutralizing antibodies can protect against the acquisition of HIV in humans. We don't have that evidence yet. We have very good evidence in animal models and certainly from laboratory assays that these antibodies are very good at blocking virus infection, but we don't know that it can do it in humans. And if this is shown to be the case, and we should know this answer in the next 2 to 3 years, this will strengthen the rationale for designing vaccines that elicit broadly neutralizing antibodies, because there's obviously a lot of effort going into trying to design vaccines that will elicit these kinds of antibodies. The other thing that it will do is it will really validate both the assay that we do in the lab, just for the aficionados on the call, called the TZMBL assay, as well as the non-human primate model or the monkey model that we use to assess antibodies. So because we've shown that these antibodies can work in laboratory assays and in animals, and if we can validate that they also protect humans, then we really have a very good model for taking things to the next level. But of course, it also establishes a platform for using antibodies for HIV prevention. Because as I mentioned, better and better antibodies are being isolated all the time. So if we can show with AMP that VRC01 is protective, it really allows us then to test even better antibodies. So that really brings me on to the story, the CAP256 story that I wanted to tell you about. It's some work that we've been involved in, and that is work that we've been doing through the CAPRISA cohort, which is based in Durban in South Africa. We can just move to the next slide. So this is just on the tip of Africa, South Africa, and the province of KwaZulu-Natal on the east coast. So CAPRISA was started, this particular acute infection cohort was started way back in 2004 by Salim Abdool Karim, who's the director of CAPRISA, and involves researchers at UCT, ourselves at NICD, and also some researchers at the University of the Western Cape. And this is a study that enrolls high-risk HIV-negative women, as I say, way back in 2004. And this cohort is still ongoing, and I'm pleased to say almost all of them are now on antiretroviral treatment. And to say that there are 2 clinical sites. There's one in Durban and a rural site in Woolen-Ledla. And on the next slide, I just have a few pictures to give you a bit of a sense of these clinical sites. So the Woolen-Ledla site, as I said, is a rural site, and the Durban site is a very high-tech, as you can see, very sophisticated clinic and laboratory infrastructure. But also just to say that many of the clinical trials also happen at these sites, and just the challenges that are faced by running large-scale clinical trials in a rural site where just even the transport issues of making sure that people can get to the clinic, which may be hundreds of kilometers away, really can be quite a challenge. Move on to the next slide. So we, as these I mentioned, have been working on, and these are in fact all women who've been enrolled onto the CAPRISA clinical or this CAPRISA study. And one of the things about broadly neutralizing antibodies is that they take a very long time to develop. So when a certain percentage of the cohort had been enrolled for 3 years, we used the stored serum samples from these individuals, and each one of those numbers on the bottom is a participant in this cohort study. we basically took their stored blood and measured it in the laboratory against a whole large panel of viruses to identify those participants who were able to neutralize a large proportion of the viruses. And what you can see is that really only a subset of people develop broadly neutralizing antibodies, and those are the ones in orange. Many people don't develop any broadly neutralizing antibodies at all, and we still don't understand why some people do. And we have some hints, but we don't know. We don't have all the answers. But I also just want to point out the CAP256, which is right on the extreme right, who really was the individual who developed the most potent and broad antibodies, and that's the reason why we have focused on that particular individual. So you move to the next slide. One thing that I often get asked, and I thought I would just include, is that the people who develop broadly neutralizing antibodies, in fact, it has no benefit to their clinical disease progression. So this shows a slide, a Kaplan-Meier curve of the time to ARV initiation, which at the time the study was done was a CD4 count of 200, or the clinical definition of AIDS. And what you can see is that the time to ARV initiation did not differ amongst the people who had broadly neutralizing antibodies and the people who didn't. And the reason for that is because in the face of a replicating virus, basically they can escape or avoid or get around the broadly neutralizing antibodies. It's a little bit like taking ARV monotherapy, which we know the virus basically gets around a single drug in the same way it can get around a single antibody. It basically escapes the broadly neutralizing antibodies. But of course we know that if those broadly neutralizing antibodies were there before somebody is exposed to HIV, these antibodies can be very protective. Well, certainly that's what we know from the animal studies. We move on to the next slide. So what we did is we teamed up with the researchers at the Vaccine Research Center to try and isolate the antibodies that are responsible for this broad activity that we were seeing in the serum of CAP256. And we were very successful in able to isolate the actual B cell and the actual antibody genes that were responsible for this potent and broad activity. And I'm just reminding you about the location of this epitope. It binds to the V2 apex, so right on the top of the trimer, which is shown there on the left. And then just to remind you as well about the potency of this antibody, so that's the red dot on the top. And then I've also just included a structure of the antibody. It is rather an unusual antibody. You can see that long loop that's sticking out at the top. It has a very long antigen. It engages with the virus through this very long binding loop. So we move on to the next slide. So one of the questions before one develops an antibody for a clinical trial is to do some preclinical studies. And is this antibody able to protect monkeys from infection? So this is a study that we did with Boris Yulg and Dan Baruch, where basically animals were injected with the CAP256 antibody, and then 2 days later they were challenged with a monkey version of HIV called SHIV. And there were 4 monkeys per group, and this antibody was tested at 3 different concentrations. And just to say that these are extremely low concentrations, I won't go into all the details but certainly the 0.08 milligrams per kilogram is not something that's been seen with other antibodies before. So if we move on to the next slide, the data from that study is shown here. So on the left-hand side is the placebo animals, so animals who did not get the CAP256 antibody, and you can see that all 9 animals had measurable viral load, so they all became infected and they all replicated virus. And then you can see on the bottom 3 graphs, the 3 different doses, 2 milligrams, 0.4, and 0.08 milligrams per kilogram. There were no infections in any of the animals. And so this really proved to us that this antibody was very effective at preventing HIV infection, or HIV infection in this case, and also that the potency that we were seeing in the lab was really translating to very good potency in an animal model, and that was also very encouraging. So we can move on to the next slide. So this is really the start of thinking about using CAP256 for HIV prevention, and this is really being spearheaded by Salim Karim, and to use broadly the CAP256 in young women for passive immunization, and basically to use bNAbs as long-acting PrEP, with the potential to impact on HIV in young women from 16 to 24 years, potentially with 3 to 4 subcutaneous doses per year. So just to say, CAPRISA is very focused on prevention in young women because that's really where the epidemic is rages the highest. And also, the SLIM is very focused on the subcutaneous route of administering the antibody. So I'm sure I don't need to remind this audience, on the next slide, I don't need to remind this audience about why we're focused on young women and focused on Southern Africa. So this is just some prevalence data showing the rate of HIV infection in young pregnant women in South Africa. And it's almost 5,000 young women, and they're broken down, as you can see, into different age groups. And just showing the prevalence, which, as you can see, is extremely high. Young women greater than 25, it's 52% of HIV prevalence. So really, this really speaks to the urgent need for having, effective prevention tools for young women in Africa. I mean, obviously, to say these are pregnant women, so these are sexually active young women in this particular region of rural South Africa. Move on to the next slide, please. And again, I'm sure you all know these data far better than I do, but this is the data showing from the Global AIDS report that we are making great progress on access to treatment. And on the left just shows the over 18 million people, and that's gone up even more since then, I'm sure, on antiretroviral treatment globally, with a third of those, almost a third of those being in Southern Africa. And I think the really depressing news is that that's had really no impact on the incidence rate, so the numbers of new infections. And that's shown on the right-hand side, where there were 2 million people infected in 2013 and then the same number in 2015. And so what that really speaks to is that we're really failing on prevention, and again, the need for us to really try some different strategies to prevent transmission of HIV. We move on to the next slide. So why do we think this CAP256 VRC26.25, I know it's a bit of a mouthful, is a candidate for passive immunization? Well, because as I've shown you, it has good coverage of Clade C. It is amongst the most potent bNAbs currently available. And something that I haven't gone too much into, I will talk about it at the end, is that it's also part of the best current combinations of bNAbs. And that's because it targets a different epitope. And so in combination, it really, its potency really provides a unique contribution to the combination. Importantly, there's no identified cross-reactivity, and that's despite the very long antigen binding arm. We haven't seen anything certainly in the lab, but it's something we obviously need to monitor in the clinical trial. As I've shown you the data, it works very well in a macaque model and animal model. The other thing, that we're excited about with this antibody is that what this will tell us about, developing a vaccine. Because if this antibody is shown to be protective, then really we would want to try and elicit these types of antibodies by vaccination, because we also know from our studies on the longitudinal and how these antibodies develop is that they actually acquire breadth much more quickly. It doesn't take the 3 years that I didn't go into those details, but actually these kinds of antibodies seem to be able to become broad much more quickly than other kinds of antibodies. And so we're also pursuing, this as a, to try and design a vaccine that can stimulate these types of antibodies as well. We move on to the next slide. So this just gives you a little bit of a breakdown of the steps that are taken in order to take an antibody all the way to clinical trial. The ticks, as I've shown, we made a lot of the antibody and did the monkey challenge studies. And we're now at the GMP, stands for good manufacturing practice. So obviously these antibodies have to be made in a laboratory that adheres to very strict quality standards. And I was very fortunate actually just 2 weeks ago to go and visit the factory that's making this antibody for this clinical trial. They make the antibody in big, what they call bioreactors. In fact, it almost looks like a brewery, these big steel chambers for making liters and liters of this antibody. And so we're at that's the stage that we're at, but obviously there's got to be some formulation so that it can be administered subcutaneously. There will be some preclinical studies, the filing of the IND, and then the phase 1, phase 2 study, which is called CAPRISA 012. So if we move on to the next slide, this is the schema that Slim has put together for the CAPRISA 012 clinical trials. As I mentioned, the focus is very much on HIV negative, and it's largely going to be young women. There will be a few HIV positive participants who will receive this antibody, just a small number. And then the sites will be the 2 sites that I mentioned earlier, Willem-Ledler site and the clinic, the eThekwini clinic in Durban. And then the study design is a phase 1 study to establish the safety and the dose, and there will be both an intravenous and a subcutaneous arm to the phase 1 studies to get the dose right. And then the phase 2, I think, is where it gets really exciting, because Slim is planning on testing this antibody in combination with 2 other antibodies, so with one other, but 2 different trials. So with the PGT121 antibody, which targets the V3 glycan, and this is an antibody that is also in clinical trial, and then also with the VRC07, which is really an improved version of the VRC01. And if these look promising, then these would obviously then go to an efficacy trial or a large-scale trial. We can go to the next slide. So this is just to tell you why the combinations are important. So on the top graph is, and this is breadth potency curves in a slightly different format, and it shows the 3 single antibodies in 3 different colors. So on the y-axis is the percentage of the viruses neutralized, and on the bottom axis is the potency of the antibody. So the more to the left, the more potent you are. So you can see the CAP256 in green. You can see is much more to the left than the others, but it doesn't reach as high as the blue one. So I'm talking about the top graph now, which is VRC07, which is much broader than the CAP256 antibody. However, when you combine these antibodies, so now we're looking at if we add 256 to the VRC07, you can see that we've now not only got the breadth, but we've now also got the potency, and the same with the combination with the PGT121, which is the red one. So we've basically shifted these curves by doing combinations of antibody and really harnessing the benefits of each antibody into the combination. And that's the explanation for that is often because these antibodies hit different viruses, and so that's why these curves shift. What I've also done here is just included the VRC01, which, as you can see in the black line, is less potent and less broad than the combinations of antibodies. So I think just in summary, if we move to the next slide, what I've shown is that, so we know that animal studies that provide a proof of concept for broadly neutralizing antibodies, but we need efficacy data in humans, and that's what the VRC01 trial is going to hopefully give us. But just to say that there are other bNAbs in clinical trials. So in addition to VRC01, there's an antibody called 3BNC117, which also targets the CD4 binding site that's been tested in humans, mostly in HIV-infected individuals, and also shown to impact on viral load. In addition, antibodies to the V3 glycan, as I mentioned, PGT121, but another related antibody called 101074 has also been shown to be antiviral in HIV-infected humans, and the prevention trials are also planned. And then the trials of the V2 glycan bNAbs. So in addition to the antibody I've just talked about, the CAP256 antibody, there's another one called PGTN1400. Both of these target the V2 glycan, and both of these trials are also being planned. But just to say that, there's broader and more potent antibodies being isolated, but also being engineered. One can also genetically engineer antibodies in the lab to make them broader and more potent, and you can also engineer them to have a longer half-life so that one wouldn't have to, continually infuse. There may be a limit to that, but certainly if we can get it out to 6 months, I think, that becomes a very feasible proposition. And then this is just really, I think still needs to be shown, but that antibodies may be more useful for prevention than treatment, but importantly, they're going to provide a benchmark for vaccines. Which brings me back, if we go to the next slide, to my starting point about, bNAbs, as I said, are in some ways a plan B, although they may end up, if they prove to be very effective, they may end up being the potential solution to the problem. But really, the reason why we really do need to continue to pursue a vaccine is because passive immunization is short-lived protection, whereas a vaccine, as we all know from many other vaccines, provides lifelong immunity in many cases. With the passive immunization, we would need to give multiple injections, whereas vaccination generally is 3 to 5 vaccinations. I guess the one real benefit of the passive immunization is that there's instant protection. And it is that, because it is essentially a biological drug, it is as soon as it's in the bloodstream, it provides instant protection. Whereas a vaccination, obviously, response would develop over months, and that's why it would be important with the vaccine that, it's given to individuals before they become sexually active. The other thing about passive immunization is that this will tell us the protective titer of this particular broadly neutralizing antibody, in this case VRC01. Whereas, with vaccination, what we are eliciting is lots of different antibodies. We elicit a whole mixture, or what we call a polyclonal antibody response. But we also elicit other immune responses. So it's a much more complex scenario, basically harnessing the immune system to protect people from HIV infection. So I think that's my last slide. I think just finally, the one last slide is just to say, the CAPRISA cohort has been, as you saw from our data, has been really a fantastic resource for us to do this sort of work, and really to thank all of the women, many of whom have been in this cohort. Some of them are from 2004, our Slender cohort, and they come for their regular visits all the time. And I think what's really exciting about this is that, we isolated an antibody from this cohort, and we're going to go back into this community and test this antibody for HIV prevention. So we're looking forward to, continuing to work with this cohort into the future. So thank you very much.
Great. Lynn, thank you so much. That was terrific. Obviously very complex, very exciting science, but you did a fabulous job in explaining it all. Lots of great questions have come in. To remind people, you can continue to email or put them in the chat feature of Ready Talk, or I'm happy to open the line in just a minute. People can hit *7 to unmute and then *6 again to mute. Lynn, a bunch of questions, and very excitingly, I think, around this idea of combinations and what comes after VRC01. So let me just throw a couple of different things at you in the interest of time and see how you best want to respond. So there were a couple of different slides you showed where VRC01 was clearly not as potent, and that presumably is why it needs to be done by infusion. But a couple of questions came in saying, well, what does it say about this study if we have all these other antibodies that seem to be better? What will the AMP study tell us? What if it's, could it be that, what happens if it's not as effective, and what does that imply for the other antibodies in the pipeline? And then by the same token, what's a way you might think about what happens if it does show high degrees of efficacy? What does it mean both for other antibodies? Does that mean they have to be even better? But also, what would come next? It's a product that is infused, and you described it, at least the CAPRISA team is looking at an injection, not an infusion. But what happens if it's very effective with VRC01? What do we do next with that?
Yeah, no, it's a great question. I mean, the reason why VRC01 is this far along is simply because it was one of the first broadly neutralizing antibodies isolated. And I mean, just to give you an idea, to make enough antibody for a clinical trial takes 2 years. So it's just a very long process. So there's no doubt that that there are better antibodies out there. But I think that what is exciting about AMP though is that it will set it's the proof of concept. I mean, VRC01 is not going to be a product, but there are better products in the pipeline, and certainly the HVTN and others, Gates and CAVD are gearing up to get better antibodies into the pipeline. And I think, the question of whether it works or doesn't work, I think the laboratory assessments are going to also be very crucial for this, because we're going to be able to detect, quite very accurately how well this worked, because we can, sequence the viruses and look at the sensitivity to VRC01. So we're definitely going to have a good idea of whether this worked or not. So I think there's a very clear signal then, obviously the next antibody would have to be assessed. I mean, I'm not quite sure about how one designs these clinical trials, but it would obviously need to be assessed against VRC01 or against some other effective prevention modality.
Great. And Lynn, you described it, and there were a lot of questions that came in about this idea of combinations, and you've described it on this call, but in previous sessions as well, about the importance of combinations, not unlike what we've seen with antiretroviral therapy, where combinations are key. How do you choose? A bunch of questions came in. You described the CAP256 being combined with a couple of different options, but I went back to a slide to put on screen for those still online. You showed these 5 different targets. How do you decide which antibody within each of the individual targets is best, and then how do you decide what to combine? Why are you combining a V1, V2 antibody with a CD4 antibody, or why not combine it with a GP120 antibody? So what's the secret in selection?
So the secret is in actually doing the assays in the lab. So we have these large panels of viruses, so we can actually test in the lab which are the best combinations. And there have been quite a number of studies that have done that, and that's actually how we came up with those particular combinations. So we can do this in the lab and basically select the best antibodies. But it's also about manufacturability of the antibodies, because not all antibodies are the same, and some are easier to make in the lab than others. I mean, some, for example, have proven to be insoluble, and so then they can't go forward for development because manufacturability is just too difficult. So there are a lot of things that go into selecting which antibodies go forward.
Great. And a couple of questions came in about related to still the discovery of these antibodies and the pricing of these antibodies. So you described very early on in one of your slides, ideally you're looking for something comparable to the cost of PrEP. So questions have come in, what might it cost to deliver an antibody? And another question came in, does a combination of antibodies automatically mean much bigger amounts of antibody and or more cost of antibody?
Yeah, no, these are all great questions. So the cost is, as I said, is a moving target, and people have done these cost analyses and claim that they can get into the range of the cost of PrEP. I mean, I think that will have to be remains to be seen. But cost should never be used as a barrier for anything, because if it works and if there's enough demand, the cost will come down. So I don't think cost should be a big factor at this stage until we. Once it works, I think that changes everything. So what was the second part of the question?
Oh, by combining two antibodies.
Oh, combining, yes. So obviously combinations will double the cost, but there's also what are called bispecific antibodies. So people can actually engineer antibodies so that they target two different things, because an antibody has two arms, and you can engineer the antibody so that one arm targets one epitope and the other arm targets another. And there's some really, I didn't get into that at all, but there's some extremely potent and broad bispecific antibodies that are coming online as well. So again, there's ways around some of these issues.
Great. So just for the lay people amongst us, so we often talk about the antibody in that Y shape, as you just said. So is it relatively accurate to say that when we talk about these combinations, you're putting two of these different Ys together, whereas what you just described, these bifunctional antibodies, that it's taking a single Y and kind of merging it from two different antibodies. That's right. Is that an accurate summary?
That's exactly right. Yeah.
Great. And a question, this idea of how potent does it have to be to move from an infusion to an injection? So I don't know if this is the best slide to show, but toward the end you were showing the combinations. And someone asked, so on this one you have at the bottom the VRC1, which is in the black at the bottom, where clearly it appears that the potency is so low that that's presumably why you need an infusion. Is there, when you talk about the sweet spot, is there also a sweet spot at which point you can move from a lengthy infusion to a subcutaneous injection? Or do we know that, or is that just an experiment with each antibody to see how you do it?
Yeah, so it'll be an experiment with each antibody, and obviously at what titer it's protective. But certainly VRC1 was, it was tested subcutaneously, but they could only put in 5 milligrams per kilogram because of it would just be too much volume. And then the other advantage of potency is that you only have to manufacture, you can manufacture a lot less antibody for the same, bang for your buck, I guess. So potency is a very important factor both for efficacy and for production.
Great. Now we have a bunch of very savvy advocates who've not only been paying attention to antibodies and vaccines, but also to PrEP. And there's a big conversation in the injectable PrEP field about what people talk about as the tail. So as a lot of people in the line know, because some of you asked the question, we have this idea of long-acting cabotegravir, the ARV, and people's concern that it might stay at suboptimal levels. Is that a concern with antibodies at all?
No, that's a very good point, because as you saw from those curves, the level of antibody drops off, and there might come a point where yes, it's no longer protective. Well, there will come a point where it's no longer protective. So that's definitely something that we're thinking about, and definitely in the same way that we have to worry about that with people coming off PrEP. It's something that has to be monitored, and people have to be made aware of that, absolutely.
And on a related note, you described, I think one of the things you're monitoring for in the AMP studies, this idea of resistance. And you were describing the resistance to, I believe, the antibody. But again, those people looking at both prevention and treatment with antiretrovirals, the idea that you might get a resistant strain of the virus and maybe not even be eligible for certain regimens of treatment. The resistance that you're describing, how different is it? Is there any reason to be worried? So if you have a breakthrough infection on PrEP, there is concern about what treatment you can get. Is that a concern at all with antibodies?
Right. So antibodies target the envelope protein, which is different. So antiretroviral drugs target the enzymes of the virus, either reverse transcriptase or protease or some of the new integrase inhibitors. So they're targeting totally different parts of the virus. So certainly there's going to be it's not going to impact on anybody's treatment. But also just to say the resistance that I'm talking about to antibodies is natural resistance. This is not induced resistance. So the sort of resistance, when we talk about antiretroviral drug resistance, we're talking about selected resistance. So people who basically are failing drug therapy and have resistant virus and then pass it along, and that obviously does compromise that person's treatment. This sort of resistance, I say, is this naturally occurring resistance. Of course, as you mentioned, though, if people are on VRC01 and the levels drop and there's some replication, we can select for antibody resistance. Then obviously the VRC01 would no longer work. But other antibodies could still be used in that case, or antiretroviral therapy. As I said, there's going to be no cross-resistance between antibodies and current antiretroviral drugs.
Great. That's super, super helpful. Another question. You had said earlier with looking at these antibodies, which is you were describing so nicely how these were discovered, that they didn't provide actual protection or therapeutic benefit for the individuals who actually have these antibodies naturally. But someone asked the question, and I'm going back to the slide where you talked about the clinical trial pathway for the CAP256 antibody, where you're enrolling, or they're planning to enroll ART-naive HIV-positive participants. So the question is, what's their role in the trial if we see this antibody not providing any protection or therapeutic benefit?
So there is a small therapeutic benefit. So a number of antibodies have been tested in HIV-infected individuals, and there is a small therapeutic benefit of. It really is 1 to 2 logs. It's not. And it is short-lived because again, virus escape happens, and again, it's monotherapy. So there is a small therapeutic benefit. But what I was showing there from the participants in the cohort is the long-term. There's no long-term benefit, but there is a short-term impact on viral load by these antibodies, and that's why these studies are important to do, to just see that it does actually have antiviral effects, direct antiviral effects. But yes, there's no long-term benefit.
Great. Now, there was a question, and I think when people, you and others have, and we have a graphic that will be included in some of the Vaccine Awareness Day materials of the number of antibodies that have been discovered, all within the last in less than a decade. So everything you've described are all brand new. And you described N6, this great new antibody. And a question about what happened all of a sudden in the last 7 years? Why are we finding them all now? And at what point do we get overwhelmed? I mean, we obviously can't test every single antibody that's discovered, but is there a point at which we say, back to your earlier slide with the sweet spot, have we found that sweet spot and we just need to now study those antibodies, or will we see more numbers and letters combined in discoveries?
No, I mean that is a great question. I guess the reason why we're finding them is we're looking harder. That's one thing, but also the techniques or the technologies have really been enabling. We know how to find them. Because these really are, it is like a needle in a haystack, trying to find the B cell that makes these potent antibodies. And the techniques have become really very sophisticated to fish them out. So it's a combination of looking harder, but also better technologies. Yeah, I mean, I guess whether there's even better and better ones out there, I mean, I guess we won't know, we need to keep looking, and I guess we will come to a point where we realize we found the best ones. I mean, N6 is getting pretty close, and so it may be that that N6, and certainly it is being planned for clinical trial, but it may need a partner, so it may need an antibody targeting another site for which we don't have the most broad and potent one available yet. So I think there is still some scope. So certain people are still isolating antibodies, and there are still some, as you saw that picture of that trimer, there was still some gray, so there may even be some antibodies targeting sites that we don't know about yet. So there's certainly still a lot of research going on in this area.
Yeah, and that's a great answer, Lynn. And I think what is remarkable, how much more we have seen not only in antibodies, but the knowledge of the virus itself just in the last few years, given technology, and yet there's still so many things we don't even know we don't know in that classic term. So, but there's so much more we do know. I know we're a little over there, but a lot of people still on and some more questions. So let me just keep going with a couple of additional ones because they're really exciting and important questions. Someone asked about the populations under study, and I know the answer to one part of it, but I'll just get to really quickly. Someone asked that you described the AMP studies, one in Africa amongst women at risk and one in the Americas with gay men and other men who have sex with men. It also includes transgender. Someone asked a question if transgender were included, and its trial is enrolling both men and transgender people who have sex with men. So I just wanted to clarify that. But related to population, someone else asked a question because there had been early talk at some of the earliest discussions with VRC01 about its use not only in adults at risk of infection, but actually as part of vertical transmission programs to prevent infection, and wondered, someone asked the question, is that still a priority given how much work is already going on successfully towards eliminating vertical transmission? So is that a priority? Should it be a priority?
Yeah, well, it certainly is still ongoing. I mean, there is a clinical trial ongoing with VRC01. I guess that's it's a different population, and there are different issues. But certainly, it still is very much under study. I think the thing about about antibodies as opposed to drugs is antibodies are natural compounds, and they also have other biological effects and over and above just their neutralizing activity. So I think people are also pursuing antibodies as part of a cure agenda to see if they can also target the latent reservoir. So there are lots of reasons to try these antibodies in different populations and different groups.
Yeah, no, super helpful. I think really, I'm so glad you made the point about the work in prevention, treatment, and cure, because I think one of the other great advances is that we're seeing research and researchers and products being studied across all three domains in very exciting ways, and the antibody piece is just one of them. Someone else has, there's obviously a lot of enthusiasm, even from those of us outside of South Africa, but certainly on your behalf and the whole CAPRISA team, and particularly the women who've been involved in those trials, that the antibody was isolated out of CAPRISA. The trials are being planned and led by South Africans, and it's a beautiful example of communities and researchers together outside of the traditional structures in the U.S. and Europe. So there were a number of people just sending congratulations on that. But questions about CAPRISA 012 in terms of the time frame of these studies. It looks very exciting to see these moving ahead with to establish safety and dosing and looking at these combinations. What is the timing of these trials, and what would and when or what would an efficacy trial look like, and when might we get some indication of that?
Yeah, so as I mentioned, the antibody is still being manufactured, but the hope is that the phase 1 trial will start a year from now, and I guess one has to wait and see how that goes. But that may take 6 months to a year, and then the phase 2 would start. So phase 3 is still a way off. But a lot of it depends on, I guess, things that could potentially slow things up, like manufacturing, like INDs. There are a number of things hurdles that still have to we still have to get through.
Sure. Nothing about this work is easy, as we all know. One of the things that someone asked, and it actually, I can't think of a better question, given that we've often thought of Vaccine Awareness Day not only to recommit and energize advocates, but some of the earliest work in HIV Vaccine Awareness Day was to thank the trial participants, because without them, none of this science happens. And so, I think very importantly, you rightly ended your presentation here. I just put the slide back up about thanking the women who participated. But someone asked the question, a lot of us have heard about CAP256 for several years. We talk about it as this number, and it's now part of all the antibody animations. There's a person behind CAP256, and someone asked, does she know? Does she know that we're all talking about an antibody that, you all scientifically isolated, but it was all because of her own body? And does she know who she is?
Right. So, obviously I don't know who she is, but certainly the clinical people do. And just to say, she's doing really well. She's obviously on antiretroviral therapy and thriving, and still comes to the clinic. And she does know, actually. She has been told. So yes, she is aware of this.
Oh, that's fantastic.
But obviously her identity is not made known to everyone.
No, absolutely. I was not suggesting we put her on a webinar unless she wanted to be. But, for those, at least in the U.S. and I think around the world, know a very important book about the secret life of Henrietta Lacks and the importance of, we can talk all we want about antibodies and manufacturing and targets and all of that, but at the end of the day, it's about individuals and the importance of their not
absolutely.
Just providing samples, but engagement. So that's great to hear. And another question just came in, and again, perhaps well-timed because of where the field is and where the world is right now. You talked a lot, and just the whole idea of your description of CAP256-VRC26, these numbers, but those names matter, CAPRISA and the Vaccine Research Center. And can you talk a bit about, outside of the actual things you're working on, the way in which this work is happening in terms of the collaborations, and is there more that needs to happen to facilitate and accelerate not just scientific discovery, but making this work more efficient across collaborative programs and sharing expertise? And what have you learned? What's working and what's not working?
Yeah, so the collaboration with the VRC has, as you can see, it's been highly successful and continues to be, actually. And one of the big things has been the exchange of scientific staff. So particularly staff from here, going over to the VRC and working there and learning new techniques, and then being able to come back to South Africa and set those up. But also, VRC people coming to the clinical site and getting involved in how one tests something like an antibody. So it's really been a very collaborative and two-way collaboration. So and of course that is dependent on funds, and so the NIH has been very generous in funding a lot of this, and obviously CAPRISA was started by an NIH grant, and so yes, it's been a highly successful collaboration and continues to be as we move towards the clinical trial.
Great. Well, it's so. This was not a setup, Lynn, but you later, early next week, before our final webinar next Thursday, we'll be releasing from AVAC an overarching kind of report on where things are in the state of HIV vaccine and antibody research, and some key messages. And I was just thinking about how, in the midst of the last hour, you have highlighted all three of those, including this last issue you just raised, and just to give you all on the line a bit of a sneak peek, and this is not going to surprise anyone, but the focus that both for HIV vaccines and the relationship to antibodies, that not only are they essential, but they're possible. And I think the work you just presented, Lynn, really shows that, although many unanswered questions, that things are possible, and that there is this energy that maintains both the importance of the research, but also the possibility of getting closer to some additional options. The second big thing, and again, you defined it beautifully with the antibody work, is progress toward an expanding pipeline. And again, just looking at this really explosion of antibodies and the combinations of them. And thirdly, and you might get a theme here, we think it's possible. We see amazing progress in the pipeline. And the third piece, the partnerships, which you just described. And so I can't thank you enough for really in the course of the presentation reinforcing those key messages that we've been grappling with internally. I think all of us working in this field, whether for weeks, months, or for 30 years, all are kind of galvanizing around. So just want to thank you so very much. I don't know if you have any last thoughts or words you want to leave this group with.
Well, no, just thank you very much for for participating and being interested in this work. That's great.
Great. Well, Lynn, thank you so much. I realize now we were quite over time, and I realized I never actually stopped because all these questions were pouring in. Maybe just since people are still online, and just any quick questions or comments from anyone on the line, you can hit *7 to unmute yourself. I would ask to introduce yourself and be very brief, but any last questions or comments for Lynn?
Margaret McCluskey, USAID, just thank you so much, Lynn and Mitchell, for such a clear web meeting, essentially, on such a difficult subject. It's so complicated, but you really explained it beautifully.
Thanks, Margaret.
Great. Thanks, Margaret. Glad you're here. And really want to thank everybody. Anybody else? *7? I did just put up this last slide. So we are 4/5 of the way there, and it won't stop on May 18th because this science and this advocacy can't stop on May 18th, and it won't stop. But I do want to just flag, of course, that all 4 webinars, including the one with Lynn, will be available or are available on the website. And I want to really encourage people for the closing webinar in the series with Nelson Michael from the Military HIV Research Program, who's going to really, hopefully, we think, pull it all together, looking at vaccine development in context, both HIV and broader vaccine development, and as the so-called vaccine hunter. So please join us, same time next week, May 18th, and we really look forward to that call. And Lynn, just thank you so very, very much for a fantastic presentation, and we will talk to you soon.
Fantastic. Thanks, everyone. Bye.
All the very best. Have great days, everybody.
Bye.